Okay. So, okay, fine. So in today's class, we are going to discuss protein tyrosine kinases, and these type of receptors, as you know, we also discussed in yesterday's class that these are also present over some kind of cells beside GPCRs. So as you can read from here, these enzyme linked receptors are second major type of cell surface receptors. Okay, so the most abundant receptors are GPCR, obviously, but after GPCR, these enzyme-linked receptors are major type of receptors which are present over the cell surfaces. And one type of enzyme-linked receptors are themselves receptor tyrosine kinases. Beside receptor tyrosine kinases, there are two more families. One is called tyrosine kinase associated receptors, and one is called receptor serine threonine kinases. Okay, so we will start with the discussion how these were discovered, what are the major advantages of their discovery in cell signaling, understanding of cell signaling pathways. So that all we will discuss in today's class. So see here, enzyme-linked receptors are the second major type of cell surface receptors, like G protein linked receptors that are called GPCRs. Obviously, they are first, they are most abundant because they were present over everywhere. Means smell, sight, taste receptors are all GPCRs. Enzyme-linked receptors are transmembrane proteins with their ligand binding domains on the outer surface of the plasma membrane. So there, where a cell is having. So let us see the structure of these receptors first, so that we are able to understand actually means. Okay, so here, so these are all receptor tyrosine kinases. Okay, so for example, this is epidermal growth factor receptor, this is insulin receptor, this is platelet derived growth factor receptors, and many more growth factors which are responsible for cell survival or cell proliferation or differentiation are coming under these receptor tyrosine kinases family. So these three major type of receptors are found, in which if you look in the first class, that is for example epidermal growth factor receptor. So here you will see there is only single polypeptide chain which is having an N terminus over the outside of the cell, and there is alpha helix. This is containing hydrophobic amino acids, and there is a C terminus which is in the cytosol of that particular cell. Okay, so this is a single polypeptide, but when ligand bind with this N-terminal domain, the receptor dimerization will result. So see here, for example, this epidermal growth factor after binding with the receptor with some growth factor or some, okay, so this will lead to the receptor dimerization. Two protein will come together and they will transmit. There is a change in conformation of these receptors so that the cytosolic domains or the C-terminal domains will be activated, and they will do cross or autophosphorylation. This is called autophosphorylation because they are doing phosphorylation over themselves. This is called cross phosphorylation because this receptor, this particular polypeptide chain is having tyrosine kinase activity, so it will phosphorylate some of the tyrosine residues in this particular chain, second chain, and this particular chain will phosphorylate the second one. So this is called cross phosphorylation. In some books you will find this particular term. Similarly, the insulin receptor is a receptor tyrosine kinase, and many times this direct question has been asked in many competitive examinations that insulin receptor is coming under which kind of family. Means so it is a receptor tyrosine kinase. You should remember this. And the second important thing which you should remember is it is having two chains, not a single chain. It is made up of two chains, two alpha actually and two beta. So one is alpha, one is beta, and there is another, there are dimers already, and these dimers are linked by these disulfide bridges. The disulfide bridges are three in number. So one is linking alpha with beta, one is linking alpha with beta, and one another disulfide bond which is present between the two alpha subunits, which is connecting these two alpha subunits so that ligand or insulin can come and bind to this N-terminal domain of alpha subunit, and the receptor tyrosine kinase activity is present in the beta subunit C-terminal domain. So you need to remember this. There are two polypeptides, one is alpha, one is beta, and already they are present as homodimers, and this signal or the insulin binding here with alpha subunit will lead to the changes in conformation in the beta subunit C-terminal domain. Okay, so there is autophosphorylation or cross-phosphorylation occurring here. Okay, so the three disulfide bases. One other family of receptor tyrosine kinases is having immunoglobulin-like domains. These are antibody-like domains, which is present, for example, in platelet-derived growth factor receptors. So, and the most important characteristic of these receptors, the third family of receptors, for example, PGDF receptors, see, focus here, it is having a blue color stretch. This particular stretch is not having any kinase activity, because this red color stretch is having some tyrosine kinase activity. But when you look into these receptors, these receptors do not have this particular blue color stretch, but this PDGF receptor, along with these IG like domains over extracellular surface, in its cytoplasmic domain or C-terminal domain, will have this particular stretch which is not having any catalytic activity. But this is a link between two catalytic domains, and this domain is this blue color stretch is made up of some 100 amino acids. Okay, so you need to remember these things from this particular classification. Now the question is how the discovery of receptor tyrosine kinases were done and how they were important to understand the overall functioning of cell signaling pathway. So now this particular experiment is very, very important. Shalu is having some questions. She is raising hand, or by mistake, I do not know. But if you have some questions, Shalu, please ask in the chat box. You please write your query. So, okay. So now these receptor protein tyrosine kinases, in contrast to when we compare them with the GPCRs, these are other cell surface receptors directly linked to the intracellular enzymes. Okay. Shreya is, please tell me. The name of blue stretch, there is no specific name to that particular blue stretch, but it is made up of some 100 amino acid residues, and it is you can say Shalu is asking why every receptor is having different type of structure. Obviously, the two humans means, Shalu, you are not common, means you are means I do not want to say, but you are not similar to any other human on earth. Okay, so every receptor is specific on its own way, and the classification is done in such a way that some of the receptors which are having the same structures are coming under the same class. Okay, Shreya is asking what the function of this stretch. Then there is no function, no non-function. Actually, this particular stretch which I showed you is just interfering with this particular stretch. Means this is not what I need to say here. This is not the single stretch. Means this kind of stretch which is present between this catalytic domain is not having any catalytic function. Therefore, it is colored with blue colored, and this red color domains. Okay, so they are having tyrosine kinase activities. Okay, Shreya. No specific name, and we will discuss. Okay, in text also it will come, but I told you before the text so that you at least have something in your mind before I tell you. Okay, fine. So besides GPCRs, this is the largest family of such enzyme-linked receptor is the receptor tyrosine kinases. We know the GPCRs are not linked directly to some of the enzyme. They are linked with the G protein, and the G protein is activating some of the enzymes, for example, adenylate cyclase or guanyl cyclase or phosphodiesterase, for example. But this is the largest family in such a sense that the receptor tyrosine kinases are interacting with some of the enzymes directly, as we will see. Okay, so which phosphorylate their substrate proteins on tyrosine residues. Therefore, they are called tyrosine kinases, because this is a special class until here actually we have studied about serine and threonine kinases, but from the today's class onwards we are moving to tyrosine kinases, and in today's class we will also discuss one specific class of enzymes which is responsible for phosphorylating not serine but either tyrosine or threonine also. Okay, but they are specific for tyrosine only. So let us discuss first about these tyrosine kinases, and then we will move to tyrosine threonine kinases. Okay, so this tyrosine kinases, a family of these kinases include the receptors for most polypeptide growth factors. So you need to underline this. Most of the polypeptide growth factors, whether it is insulin or epidermal growth factor, pellet-derived growth factor, and so on and so forth, many were discussed in yesterday's class also. Example are given here: transforming growth factor, these fibroblast growth factor, colony stimulating factor 1, all are coming under this receptor tyrosine kinases. Okay, and they are having an intrinsic enzyme activity, or they are associated directly with an enzyme. I will tell you this thing also. So please have patience and we will move step by step. So there may be two possibilities. Either they are having this intrinsic enzyme activity in their C-terminal domain. See here. Okay, so this is a receptor tyrosine kinase. So this red color stretch is a receptor tyrosine kinase. So it is catalytically active domain, C-terminal domain, because it is C-terminal domain of this particular polypeptide or this receptor tyrosine kinase. In some cases, okay, so when actually this is the receptor tyrosine kinase, this red color portion. So either they phosphorylate each other over this catalytic domain so that this catalytic domain activity will increase, or they will phosphorylate each other outside this particular red color stretch, that is the blue stretch actually, which is not having any catalytic activity but have some tyrosine in this particular region. And when this blue color stretch tyrosine is phosphorylated, it will create the binding site for some of the proteins, downstream signaling molecules or proteins or enzymes, which are having this particular domain, this SH2 domain. What is the name of this SH2? This is SRC homology domain 2. So either this SH2 domain or the one another domain we will discuss in the text. So how this particular domain and all these things were discovered, I will come to that portion. But before starting that portion or that experiment, I would like to tell you either this particular receptor tyrosine kinase is having a kinase activity, or they are not having this particular red color stretch. Okay, this is also possible in some cases. So what is the possibility if it is not having this particular red color stretch? Then see here, then for example, this particular receptor tyrosine kinase, for example, these are cytokine receptors which are not having this particular red color stretch, or means they are not having any tyrosine kinase activity in inbuilt. Okay, so these are no intrinsic activity, but here the cytoplasmic C-terminal domains are interacting with some other proteins or enzymes which are called non-receptor tyrosine kinases. Means these are not the part of these receptors, but they are having tyrosine kinase activity. So they are interacting with these domains, C-terminal domains. Hope this is clear. This is not rocket science. This is very simple to understand, that these type of receptors which interact with cytokines do not have the red color stretch, so therefore they cannot function. Okay, so therefore they cannot function. Therefore they require some additional enzymes or proteins so that the receptor tyrosine kinase activity can be restored. So in this case, these proteins which are interacting with these receptors will lead to the phosphorylation of some of the tyrosine over these receptors. Okay, so that they can phosphorylate each other. Also, they are able to phosphorylate the C-terminal domain tyrosine residues. So here it is having some tyrosine, here it is having some tyrosine. So these phosphorylation tyrosines, then the downstream molecules will come and they will bind with these phosphotyrosine residues with the help of their SH2 domain. So it is very important, these proteins which are having these SH2 domains will only interact with these phosphotyrosines or these receptor tyrosine kinases. So you need to remember this, that the specificity is one of the major characteristic feature of signal transduction. There should be very specific, otherwise God knows what will happen inside the cell. So they are very specific. Okay, so specific interaction always happen. Now, so these two scientists, one is called Tony Hunter and second is called Dr. B. Safton. So these two were responsible for their paper. They have presented their result in this headline, transforming gene product of Ross sarcoma virus phosphorylate tyrosine. And this discovery around 1980s was very major discovery of that time, because everyone only have knowledge about serine and threonine kinases, and only these two scientists tell the community that there is a possibility that these tyrosine kinases may be present within the cells and they will do another type of cell signaling. Therefore, this class is very important from this point of view. Okay, so actually, Rous sarcoma virus was isolated and known since 1911. People know about Rous sarcoma virus and became the first virus that was generally accepted to cause the tumors in animals. So I want to show you a new screen. See here. So this is Rous sarcoma virus. Okay, so this Rous sarcoma virus was known since 1911. Okay, and if you want to read about this particular virus, so it was discovered by, okay, so Peyton Rose in Rockefeller University, New York City, and he was doing some experiments over chicken tumor, and it was known since that time that it is having some genes, GAG, POL, EMV, and this particular gene is very important from this point of view, SRC gene for Rous sarcoma virus. So the sarcoma virus gene encodes a tyrosine kinase that attaches phosphate group over the amino acid tyrosine in the host cell proteins. This was not known in 1911. It was discovered later, but now it is known that this particular SRC gene is encoding a protein, and this particular protein is a tyrosine kinase. And this particular tyrosine kinase is responsible for causing the cancer or tumor in the, okay. So the experiment what this Peyton Ross was doing, he was infecting some of the chickens with this particular Ross sarcoma virus. Okay, so Ross virus, because the Ross has discovered this particular virus, infecting the chickens, and it was showing the cells were showing the uncontrolled proliferation. These uncontrolled proliferations are coming from the receptor tyrosine kinase activity of this particular virus. Okay, so we were, okay, so Rous sarcoma virus discovered in 1911, and it was known that it is causing some tumors in animals. Several features of Rous sarcoma virus then made it an attractive model for studying the development of cancer. Now, with time, it is coming more and more popular. And okay, Ankita is asking, sir, SS2 have any specific function? Okay, so obviously it is having function in those proteins in which this particular SS2 domain is present. They are able to bind with this phosphotyrosine. Okay, Ankita. So it will help the protein to bind or to interact with phosphotyrosine or the receptor tyrosine kinase actually, so that the signal can transmit downstream the cell. Okay, so this Rous sarcoma virus will have become a major model to study the cancer actually. Screen is paused. Okay, so we are coming back to Cooper, and in particular, in the small size of Rao sarcoma virus genome offered the hope that because this is having very small genome, okay, so it will create a hope for the scientists so that the identifying the specific viral genes, out of which one was SRC gene, which is encoding for that particular receptor tyrosine kinase. So they want to know how this particular virus is able to do this particular abnormal proliferation in the cells to create a tumor or cancer that is characteristic of the cancer cells. This goal was raised in 1970s, took so many years. Okay, when it was established that a single Rao sarcoma viral gene called SRC for sarcoma, so three letters has taken, so it is called SRC for sarcoma, SAC, is required for the tumor induction. Importantly, a closely related SRC gene was also found to be part of normal genetic complement in the variety of vertebrates, including the humans. So, in fact, in humans also, a closely related gene has been identified. So, it is very closely related to this particular SRC gene, which is a gene of this particular virus, Rao sarcoma virus. Now, since the viral SRC protein is responsible for driving the uncontrolled proliferation of these cancer cells, it appeared that understanding SRC function would yield crucial insight into the molecular basis of both the cancer induction and regulation of the normal cell proliferation. So, scientists want to achieve two targets simultaneously. First, they want to understand about the molecular basis of cancer induction, how this SRC gene is able to induce the cancer in normal animals. Okay, and regulation of normal cell proliferation, because a similar gene was found in humans also, in other animals also. So they want to know how the regulation of normal cell proliferation is achieved within animals. So after 7 years, when this was established that this particular gene was identified, another scientist, Ray Erickson, and his colleagues identified SRC protein. So from the animals, they were able to isolate the protein, SRC protein. So first the gene was discovered, and in 1977, Ray Erickson was able to isolate the SRC protein. So immunoprecipitation actually. So one technique called immunoprecipitation. So it is obvious. Immuno, what is the meaning of immuno? So we are using antibodies, or okay, so we are using antibodies to that particular protein, SRC protein, and with the help of these antibodies we are able to remove the SRC protein from the animal. Okay, so with anti-sera or antibodies from the animals bearing this Rao sarcoma viral induced tumors. So from those animals which are infected with the Rao sarcoma virus, so they will have tumors, and from that particular tumor cells, we can make the antibodies for against those particular tumor cells, so that they are able to bind to this particular protein, SRC protein, and they can be isolated. Okay. Shortly thereafter, it was found that incubation of SRC immunoprecipitate with the radioactive ATP resulted in phosphorylation of immunoglobulin molecules. थोड़े ही टाइम बाद साइंटिस्ट को पता चला कि अगर एटीपी को रेडियो लेबल्ड करते हैं और इस पार्टिकुलर रेडियो लेबल्ड एटीपी के साथ में इस इम्यूनोप्रेसिपिटेट को, जिसको अभी इन लोगों ने रे एरिक्सन और उसके कॉलीग्स ने आइसोलेट करा था, अगर उसको रेडियो एक्टिव एटीपी के साथ में incubate कर दें, तो it will lead to the phosphorylation of antibody molecules or immunoglobulin molecules. So, and it was not recognized until here that this particular phosphorylation is over tyrosine residue, because we know these immunoglobulins or antibodies are glycoproteins. So they were made up of carbohydrates and proteins, means amino acids. So these amino acid tyrosine will be phosphorylated by this radio labeled ATP. So how it was achieved? So we'll see. SRC therefore appeared to be a protein kinase clearly implicating the protein phosphorylation in the control of cell proliferation. So इससे ये पता चला कि एक्चुअली ये जो प्रोटीन फॉस्फोराइलेशन हो रहा है या प्रोटीन्स को या इम्यूनोग्लोबुलिन्स एंटीबॉडीज को फॉस्फोराइलेशन हो रहा है, ये कहीं ना कहीं से तो इसका रोल है. इट इज कंट्रोलिंग द सेल प्रोलाइफरेशन and is responsible for uncontrolled proliferation so that it was able to create tumor in animals. So all previously studied protein kinases phosphorylate serine or threonine residues. अब से पहले जितने भी हमें पता थे वो सभी kinases क्या थे, या तो serine को phosphorylate करते थे या threonine को phosphorylate करते थे, which were also only phosphoamino acids to be detected in animal cells. इसलिए हमें केवल, और दूसरी बात, उस टाइम तक साइंटिस्ट को इन्हीं दो फॉस्फो अमीनो एसिड्स की नॉलेज थी. दे वर थिंकिंग लाइक या तो फॉस्फोसीरीन होता है या फॉस्फोथीरियोनिन होता है, क्योंकि एक दूसरा रीजन था, दे वर नॉट एबल टू आइडेंटिफाई द फॉस्फोटायरोसिन दो इट वाज प्रेजेंट विद इन द नॉर्मल सेल्स आल्सो, बट द कंसंट्रेशन वाज वेरी लो. I will tell you exactly what is the concentration in the normal cells. It is just the concentration of CO2 in our environment. Okay. So, however, Walter Eckert and Tony Hunter, the same scientists which have discovered the receptor tyrosine kinase, along with another scientist, Walter Eckert, have observed in 1979 that oncogenic protein of another animal tumor viruses. So they were working over another viral model, that is polyoma virus. Now see what is polyoma virus first. So we are moving to this particular page. See, so this is polyoma virus. So this is completely different from raw sarcoma virus. But what is common? So this polyoma virus is also able to induce tumor in animal cells. So, okay. Virus whose natural hosts are primarily the mammals or birds. So, okay. So the Tony Hunter and the other scientists, Eckhart, were actually working over this polyoma virus. And from this polyoma virus, they got an idea that it is possible that the same thing can happen in the raw sarcoma virus also. And they were so much confident over this phosphotyrosine or this tyrosine kinase activity of these receptors that they already have an idea before doing the experiments. Okay, so these two scientists were working over this model polyoma virus in 1979 and was phosphorylated over a tyrosine residue, and they are getting results that this tyrosine residue was phosphorylated. Hunter and Safton therefore tested, then these two scientists came in picture, one is Hunter, one is Safton. These tested the possibility, they are having a possibility already in their hands that SRC, this particular protein, this particular gene, sarcoma gene, therefore tested the possibility that SRC might phosphorylate the tyrosine. So it will also phosphorylate the tyrosine if it is not phosphorylating the serine or threonine. So rather than serine and threonine, residues in the substrate proteins. So their experiment demonstrated that SRC does indeed function as a protein tyrosine kinase. And finally, with the help of some experiments, they are able to establish it with some very simple experiments. So I will tell you what they did actually. So this activity now recognized as playing central role in cell signaling pathways, because we know today most of the growth factors are working by this mechanism of tyrosine phosphorylation. So, okay, so we will come here what was the experiment they did actually. So the Hunter and Safton identified the amino acid phosphorylated by the SRC protein from raw sarcoma virus by incubating the SRC immunoprecipitate with radioactively labeled P32 labeled ATP. So this question can be asked. So this is P32 labeled ATP. So if the ATP is radio labeled, so the kinase will transfer this gamma phosphate from this particular ATP so that the tyrosine will be phosphorylated. Okay, so the amino acid that was phosphorylated by SRC in the substrate protein, in this case is immunoglobulin antibodies actually, therefore became radioactively labeled with the P32. The immunoglobulin was then isolated and hydrolyzed. Then they isolate these antibodies and hydrolyze these antibodies into give amino acids. So you have studied many ways of hydrolyzing proteins. For example, C-terminal sequencing, N-terminal sequencing, and many other. We know today there are many proteases which cleave the proteins or these antibodies at specific sites so that the amino acids can be isolated. So in 1980s, they isolated all the amino acids from these antibodies and they do some of the chromatography and electrophoresis experiments. With the help of these methods, separated the phosphotyrosine, phosphoserine, and phosphothreonine because they know other amino acid cannot be phosphorylated. So they are having the possibility of phosphorylating these three amino acids only. So, and this have seen that there is nothing was obtained at the sites where the serine should come or threonine will come, but they have seen a major phosphorylation is over the tyrosine only. So this P is radioactively labeled P32 actually, phosphate. I hope you understand the crux of this particular experiment. And if you have some doubt, you please read this experiment. And okay, so identification of phosphotyrosine and immunoglobulin phosphorylated by this SRC protein and an immunoprecipitate containing the sarcoma virus. SRC was incubated with this particular radioactively labeled ATP, and then they were identifying it with the help of some X-ray film. These broken lines, these are broken lines actually. So broken line indicate the positions of unlabeled phosphoamino acids which were actually used as markers. Note that the principal P32 labeled amino acid is phosphotyrosine. Now, what is the impact of identifying this? And before telling you the impact, this is given here in normal cells. Actually, why these scientists were not able to detect the tyrosine, because the phosphotyrosine accounted only about 0.03% of total phosphoamino acids which are present in a normal cell, explaining why it was not previously identified or escaped the detection actually. However, phosphotyrosine was about 10 time increase, more abundant in the cells when it is infected with this particular virus, Rouse sarcoma virus. So obviously showing this that the tyrosine phosphorylation is increased over 10 times when this infection is happening. So that's how it is able to create the tumors in animal models, suggesting that an increased protein tyrosine kinase activity of the viral SRC protein was responsible for its ability to induce the abnormal cell proliferation and causing the tumors. Now, what was the impact of this discovery? So the discovery that the SRC was a protein tyrosine kinase both identified a new protein kinase activity and established this activity as being related to the control of cell proliferation. So it is going to control the cellular proliferation somehow. That's why they are doing some, because it is already present in normal cells also. So it is doing some cell proliferation control activity, but when it is in abundance, means 10 times increase, it will lead to the cell divide uncontrolled manner, in an uncontrolled manner, so that the tumor will result. So there is a link, there is a generalization of link between the protein tyrosine phosphorylation and abnormal proliferation of cancer cells. Another scientist, Stanley Cohen, and his colleagues then identified an epidermal growth factor receptor is a protein tyrosine kinase. And after that, many other receptors, for example, insulin receptor and platelet-derived growth factor receptors, these were identified and classified according to their structures. Okay, so, continuing studies have identified numerous additional receptors, non-receptor protein tyrosine kinases. For example, these are those receptors which are not having the inbuilt tyrosine kinase activity in their C-terminal domain and are interacting with some of the tyrosine kinases. Okay, so that function in a variety of cell signaling pathways. For example, this is generally in the growth factor receptor pathways. The non-receptor protein tyrosine kinases are cytokine receptor pathways. So cytokines, for example, erythropoietin hormone, okay, so which help in the generation of RBCs. Interleukin 2, which help in the generation of T cells. Okay, so these are non-receptor protein tyrosine kinases. that function in a variety of cell signaling pathways in a variety of ways. We will discuss the ways in which these receptor tyrosine kinases function also. So there are three ways actually by which these lead to the activation of downstream effector molecules. Studies of mechanism by which the virus can cancer in the chickens thus revealed the previously unknown enzymatic activity that plays a central role in signaling pathways that regulate animal cell growth, survival, and differentiation also. And if you want to design a drug against these cancer or some tumor cells, so these are the best targets. Okay, so the specific drugs which are, if you need to design against cancer cells, then you need to target the protein tyrosine kinases because these are implicated in the tumor proliferation actually. So this may be a question. Okay, so majority of the, because polyoma or from raw sarcoma virus, it has been recognized that majority of the tumor causing viruses are receptor tyrosine kinases. So you can have a common sense that a drug can be designed against these receptor tyrosine kinases to control the unregulated proliferation of cancer cells. So these are very hot targets actually to design a drug against cancer cells. Now, indeed, the first tyrosine kinase was discovered in 1980s by these two scientists, Hunter and Safton, during studies of oncogenic protein or animal tumor viruses, in particular this Rao sarcoma virus by Tony Hunter and B. Safton. Then this EGF was discovered and found to be a receptor tyrosine kinase by Stanley Cohen and his colleagues, clearly establishing now, because after so many discoveries, it was clearly established that the protein tyrosine phosphorylation is a key signaling mechanism in response to the cells, in response to the growth factor stimulation. And now it is time to count the number of receptor tyrosine kinases. So as I told you when we were studying the GPCRs, and the last page, the mammalian genome encode about 20 different alpha subunit, 5 beta subunits, and 12 gamma subunits of G proteins. Okay, so similarly, the human genome encode for about 59 receptor protein tyrosine kinases, including the epidermal growth factor, nerve growth factor, platelet-derived growth factor, insulin growth factor receptor, and many other growth factors actually. So most of the protein or peptide growth factors or hormones are acting by these protein tyrosine kinases. All these receptors share a common structural organization. N-terminal domain is extracellular, which will bind to the ligand, and a single transmembrane alpha helix, which will traverse your plasma membrane, and a cytosolic C-terminal domain with which is having either a protein tyrosine kinase activity or interacting with some protein tyrosine kinase protein. Okay, most of the receptor tyrosine kinases consist of single polypeptide, although insulin receptor and some other are made up of dimers actually. So they are two. So we have discussed this thing also. So these are related receptors or dimers. For example, this insulin receptor made up of two polypeptide chain, alpha and beta, are connected by sulfide bridges, three disulfide bridges. So binding of the ligand with, the, for example, growth factor to extracellular domain of these receptors activate the cytosolic kinase domain. So they are all have this cytosolic, this extracellular N-terminal domain where the growth factor will bind. Okay, here, or here. Then they're having a C-terminal cytosolic domain, which is having some catalytic activity, resulting in phosphorylation of both the receptors themselves and the intracellular target proteins. Now, what is the benefit? Why you need to phosphorylate these portions? Why you need to phosphorylate the catalytic domains? So by phosphorylating these catalytic domains, so there is an increase in catalytic function of these domains actually. And why we need to phosphorylate the extra catalytic domain parts, these are blue color stretch tyrosines, because these will create the binding sites for the downstream effector molecules for proteins which are having SH2 domains. Okay, so both are having their own importance. So now, So we have discussed this, that epidermal growth factor is a monomer, but when the receptor dimerization will result after binding with the after binding with the ligand. So the interaction between the different receptor polypeptides. Now there is two things. One is called SH2 domain and one is called PTB domain, for phosphotyrosine binding domains. So these two type of domains, or the protein which are having these two type of domains, can interact with these phosphotyrosine residues over this particular receptor, cytosolic side. Okay, so now we will discuss this. Ligand induced dimerization then lead to the autophosphorylation or cross phosphorylation of the receptor, as the dimerized polypeptide chain cross phosphorylate each other or one another. Such autophosphorylation plays the two key roles in signaling from these receptors. The first is phosphorylation of tyrosine residues within the catalytic domain, which I have told you in the catalytic domain. First, they need to phosphorylate it in this red stretch region. What is the importance? It is given here. Okay, firstly, the phosphorylation of tyrosine residue within the catalytic domain increases the protein kinase activity. So you need to underline this. They will increase this kinase activity. Second, the phosphorylation of tyrosine residue outside the catalytic domain create a specific binding site for additional protein which are having either SH2 domain or PTB domain, so that they can bind with them specifically, that transmit the intracellular signal downstream of the activated receptors. The association of these downstream signaling molecules with the receptor protein tyrosine kinases is mediated by these protein domains that bind to the specific phosphotyrosine containing peptides. Okay, so these domains are characterized by having these SS2 domains for SRC homology 2 domain, because they were initially recognized in the protein tyrosine kinases related to SRC. So the kinases which are related to these SRC protein from the Rao sarcoma virus, they are very related to this, so they are named accordingly. So SS2 domain containing proteins. SS2 domain consists of approximately 100 amino acid residues and bind to specific short peptide sequence containing the phosphotyrosine residues. So they will specifically bind with this particular amino acid if it is phosphorylated. Other protein bind to the phosphotyrosine containing peptide via this PTB domain. This is phosphotyrosine binding domain. The resulting association of SS2 or PTB containing proteins with activated receptor tyrosine kinases can have several effects. First of all, it will localize to these proteins to the plasma membrane. Suppose some protein is present in the cytosol, so after binding with this phosphotyrosine, they will be recruited towards plasma membrane, so they will be localized here. Okay. Okay, so it is helping in the localization of protein, promoting their phosphorylation and stimulate their enzymatic activities. Once they're phosphorylated, they will be activated and their enzymatic activities will be transmit the signal downstream. So the association of these proteins with autophosphorylated receptors, this represents the first step in intracellular transmission of signals initiated by binding the growth factor to the cell surface. Suppose some signal will bind to the cell surface. See here, so here with the cell surface, this growth factor will bind to the N-terminal domain. Now there is a conformational change and phosphorylation of these two type of tyrosines present either in the catalytic domain or outside the catalytic domain. Then this particular protein will bind to this particular phosphotyrosine, so that it will be phosphorylated. Okay, because this is a tyrosine kinase again. So it will be phosphorylated and then it will be activated. This will be activated and this will transmit the signal downstream, so that it will phosphorylate or activate some other proteins in the cytosol, so that the signal will transmitted from here inside the cell to its target. Okay. So a model has been given. So it is showing the amino acids which are interacting with the phosphotyrosine. Actually, this is the SS2 domain of the protein. It will interacting with phosphotyrosine. So it is shown as space filling model and written here somewhere. Okay. This I already told you that there are certain receptors, for example, cytokine receptors, which are, for example, interleukin 2 receptor, erythropoietin receptor, or some polypeptide hormone, for example, growth hormone. Okay, so these will not have a catalytic domain inside the cytosol. but they will interacting with some other proteins. So these are called non-receptor tyrosine kinases, which will help in phosphorylating and creating the phosphotyrosine residue, so that the SH2 containing proteins, or uh, what was that, PTB containing phosphotyrosine domain containing protein can bind to the, and downstream effector molecules can bind to these phosphotyrosine residues. So okay, so they will function analogously to the receptor tyrosine kinases. So, and will be discussed in the previous section. Now there is a kinases associated with cytokine receptor belongs to the, now the question is, this is something different, because these are cytokines. So genus kinase will come into the picture. The genus kinase family, the kinases associated with cytokine receptors. These kinases are from the genus kinase family, which consists of four related non-receptor protein tyrosine kinases. Members of genus kinase family appear to be universally required for signaling for cytokine receptors. जहाँ भी cytokine receptor की बात आएगी वहाँ पे genus kinase present होगा. So genus kinase are very closely related to these cytokine receptors. Okay, universally present, indicating that genus kinase family kinases plays a central role or critical role in the coupling these receptors to the tyrosine phosphorylation of the intracellular target proteins. Additional non-receptor protein tyrosine kinases belong to the SRC family. Now it is SRC, so this is the same Rao sarcoma virus protein. Okay, so wait a minute. So if you are having some queries, so it is time to ask your queries before we switch to other page. And it is a break for you for 2 or 3 minutes, so we will start again. Okay, so there are no queries. Okay, so fine. Additional non-receptor tyrosine, protein tyrosine kinases belong to this SRC family, which consists of SRC and 8 closely related proteins. As already noted, SRC was initially identified as the oncogenic protein of sarcoma virus and was first protein found to possess this protein tyrosine kinase activity. So it has played a pivotal role in experiments leading to our current knowledge, understanding of the cell signaling. Okay, so members of the SRC family plays a key role in signaling downstream of the receptor tyrosine, protein tyrosine kinases from antigen receptors, for example, B and T cells. Okay, so now this is immunology portion. So B and T cells, with respect to these immuno, so for example, with response to this cytokines, will be activated. Okay, discussed later in this chapter from integrins that resides in cell attachment to extracellular matrix. Also, the cells which are interacting with the extracellular matrix are these SRC family members. Okay, so they are controlling the cellular proliferation, and if the tyrosine phosphorylation is in excess, it will lead to uncontrolled proliferation of those cells and creating some tumor-like condition. Now, there are some receptors which are linked to other enzymatic activities, so we need to discuss these also. Okay, so these are rest we have discussed actually. So although there is a vast majority of enzyme-linked receptors stimulate the protein tyrosine phosphorylation, some receptors are associated with other enzymatic activities. Okay, so these are including protein tyrosine phosphatases. Okay, so if you did a lot of phosphorylation of over tyrosine, so you need to now remove the phosphatase, will remove those phosphate groups over from those tyrosine so that a balance can be created. Okay, so the protein tyrosine kinase these protein tyrosine phosphatases remove the phosphate groups from the tyrosine residues. This will counterbalance the effect of protein tyrosine kinases. In many cases, these protein tyrosine phosphatases plays negative regulatory roles over in cell signaling pathways by terminating the signal. So they will help in terminating the signal so that a signal will come so that will help the cell to proliferate in an uncontrolled way. So it will cancel that particular signal. So the protein tyrosine phosphatases are required. There is protein serine threonine kinases and guanylyl cyclases. So, however, some of the protein tyrosine phosphatases or cell surface receptors whose enzymatic activities plays a positive role in cell signaling. For example, 21 such receptors, protein tyrosine phosphatases, are encoded in the human genome. So one thing you know, there are about 59. Okay, so I don't know why this is not working. So there are some 59. So these were encoding the receptor tyrosine kinases in human genome, and these are versus 21 means okay, so these 21 are encoding for phosphatases, whereas 59 are encoding for kinases. So they are counterbalancing each other. So they are counterbalancing each other. So they will add phosphate groups over tyrosine residues, and they will remove these phosphate groups from these. Okay, so now 21 such receptor tyrosine phosphatases have encoded in the human genome. A good example is provided by CD45. So you need to remember the example is CD45 is a protein tyrosine phosphatase which is expressed over this B and T cell in lymphocytes. Following antigen stimulation, CD45 dephosphorylate a specific phosphotyrosine that inhibit the enzymatic activity of the SRC family member. The CD45 protein tyrosine phosphatase act as to stimulate the non-receptor tyrosine kinase activity. So, okay, it is somehow activating this particular kinase. Okay, and the CD45 itself are protein tyrosine phosphatase. Okay, another thing is the cytokine receptor are used by HIV virus as a cell surface receptor to infect the T helper cells. We know this is doing some infection in T helper cells, and these are infected with this HIV virus. Okay, so with the help of these cytokine receptors, CD4 receptors. Now, the second class is these are after this there is protein serine threonine kinases. So if you ask me a question whether all these receptor tyrosine kinases are acting by tyrosine phosphorylation or there is some other possibility, so I will say there is a possibility of phosphorylating the serine or threonine residues also, or there are guanyl cyclases, so they will produce cyclic GMP as a secondary messenger. So now we will discuss about transforming growth factor and transforming growth factor receptor, and related polypeptides are protein kinases. These are kinases, but they will phosphorylate either serine or threonine rather than a tyrosine residue on their substrate proteins. So now this transforming growth factor beta is a prototype of family of polypeptide growth factor that control proliferation and differentiation of a variety of cell types. Cloning of the first receptor for member of TGF beta family is done in 1991, revealed that it is a prototype of a unique receptor family with a cytosolic protein serine threonine kinase activity. So it is having a domain which only phosphorylate either serine or threonine, not the tyrosine actually. Since then, receptors for additional TGF beta family members have been similarly been found to be protein tyrosine kinases. उसके बाद से जितने भी डिस्कवर हुए वो सभी serine threonine kinases ही थे. Binding of ligand to these receptors results in the association of two distinct types of polypeptide chains, which are encoded by different members of TGF beta receptor family to form a heterodimer. So you need to remember this. They are not forming homodimers. In this case, they will form heterodimers, and you need to underline this heterodimers in which one of the receptor kinases phosphorylate the another. Okay. The activated TGF beta receptor then phosphorylate members of family of transcription factors, and these transcription factors are called SMADs. Okay, so you also need to remember this class SMADs. We will discuss about SMADs in our upcoming classes. Okay, so these SMADs are transcription factors. So they will go into the nucleus. They will control this. They will go into the nucleus, translocate in the nucleus, and stimulate the expression of the target genes, with the genes which are under control of these transcription factors. So transforming growth factor beta is a kinase which will phosphorylate either serine or threonine. They are activating some of the transcription factors called SMADs, and they are controlling the gene expression. Okay. So if from here we are able to understand that not all cell proliferation and differentiation controlling kinases are tyrosine kinases, but some of them, for example, transforming growth factor beta receptors or the related ones are serine or threonine kinases. Okay, so they are not tyrosine kinases again. Then there is a third class, which are guanyl cyclases. So some peptide ligand bind to the receptors with these receptors, for example, receptor tyrosine kinases, whose cytosolic domains are guanyl cyclases. So there is a possibility that some peptide ligand, a signal molecule, will bind to these receptors, which are whose cytosolic domains are a guanyl cyclase, which catalyze the formation of cyclic GMP from GTP. पहले भी हमने डिस्कस करा था कि नाइट्रिक ऑक्साइड की साइक्लिक जीएमपी को एक्चुअली साइक्लिक जीएमपी के थ्रू अपना फंक्शन शो करता है, बट it will decrease the concentration of cyclic GMP. It will activate some of the phosphodiesterase enzyme, and it will cleave the cyclic GMP to GMP. Nitric oxide also act as by stimulating the guanyl cyclase. Okay, but the target of nitric oxide is an intracellular enzyme. This intracellular enzyme is a phosphodiesterase. Okay, so if you forget the nitric oxide signaling, so I will lead you to another PDF so that you can understand this better. So see here. So it will given here actually. So NO is going to activate a guanyl cyclase, which will create cyclic GMP, and this cyclic GMP will also activate the protein kinase G. Okay, so we have discussed protein kinase A, we have discussed protein kinase C. Now there is another type of protein kinase that is called protein kinase G. and fourth one is protein kinase B. So, okay, so in today's class, we will discuss about protein kinase B, and we will discuss about protein kinase G in some other class. Okay, so we are leaving this, and we were here. I do not want to mix the things so that it will create some hindrance in your understanding. So therefore, I'm going step by step only. So this we have discussed actually. So where we were actually. Okay, here. So these guanyl cyclases, okay, the ligand binding stimulates cyclase activity leading to the formation of cyclic GMP. A second messenger whose intracellular effects are discussed in the next section of the same chapter. Other receptors bind to the cytosolic proteins with additional biochemical activities. For example, tumor necrosis factor, it is a cytokine induces cell death, perhaps as a way of eliminating the damaged or unwanted cells from the tissues. Tumor necrosis factor receptor and related death signaling molecules are associated with specific proteases. So actually, these are the fourth category in which these cytoplasmic proteins are interacting with some of the proteases, so which are activated in response to the ligand binding, for example, tumor necrosis factor. So when it binds to its receptor, it will activate these proteases, and these proteases will degrade various components of the cell. Activation of these receptor-associated proteases trigger the activation of additional downstream proteases, ultimately leading to the degradation of a variety of intracellular proteins, and it will lead to the death of the cell. Then this we have studied already, because this adenylate cyclase is creating cyclic AMP, and phosphodiesterase is going to remove this cyclic AMP from the cells. Protein kinase A we have discussed, and so many things, CRAB, CRE, all these things have been discussed in earlier classes. Yes, this we have studied yesterday about phospholipids, phospholipase C, phospholipids, and calcium ion signaling along with IP3 DAG pathway. Okay, calcium calmodulin pathway is also I have discussed yesterday. Then there is ryanodine receptors, which we have discussed in our earlier classes. Means this the receptors which are present in a ER membrane, which are the gate for calcium ions to come out in the cytosol. Now we need to discuss PI3 kinase, AKT, and mTOR pathway. But actually, one more pathway we need to discuss by which these receptor tyrosine kinases will act. So, okay, so now we will switch to the Pathfinder. Okay, how many students? Only 6 students are there in the class. But I hope these are regular students, so they are able to understand whatever we are discussing. See, so receptor tyrosine kinases, all receptor tyrosine kinases comprises of an extracellular domain and terminal domain. Okay, so containing a ligand binding site, a single hydrophobic alpha helical transmembrane domain. This particular portion is alpha helix, and a cytosolic domain that include a region with protein tyrosine kinase activity. So this is having some kinase activity or catalytic site. The red color stretch actually shown here in the form of box. Okay. So with the help of ATP, they will transfer the gamma phosphate from ATP to the tyrosine residue. So they will do autophosphorylation or cross phosphorylation of these domains, and then they will do some phosphorylation outside this particular catalytic domains, so that they can create binding sites for some of the proteins which are having either SS2 domain or PHB domain, phosphotyrosine binding domain. What was that? Sorry, I missed. It is PTB domain actually, protein tyrosine phosphotyrosine binding domain. Okay, so actually I forgot, sorry. Now the most receptor tyrosine kinases are monomers. For example, epidermal growth factor receptor is a monomer, as we have seen. The ligand binding induces the receptor dimerization, so it will form a dimer. Okay, so actually in this case, it is not shown in the correct way. Uh, okay, so as we have seen from the Cooper, so see here, so the same growth factor will be binding and enabling the two receptors to form a dimer, but here this is not correctly shown. Okay, so it is something different, but somehow they will helping the two receptors so that the dimer will form, receptor dimerization will happen. And this receptor dimerization then induce some of the conformational changes so that these catalytic domain can phosphorylate each other. So the resulting phosphotyrosine functions as a docking site where these docking sites will act as signal transduction proteins, will interact with some of the other proteins which are having either SS2 or PTB domain so that they can transduce the signal within the cell. Now the question is, what are the ways by which these are able to transmit the signal inside the cell? So the two type of regulation means this particular regulation we have studied yesterday. This is called IP3 DAG pathway. And IP3 DAG pathway is common whether it is in GPCR signaling or in this receptor tyrosine kinase signaling. This is common. So there is formation of two secondary messengers. One is called IP3, one is called disylglycerol. Disylglycerol will interact with the cell. IP3 will go to the endoplasmic reticulum where they are having ryanodine receptors, able to bind to these ryanodine receptor and will release the calcium ions so that the calcium ion concentration will increase in the cytosol. About 10 time increase will happen. And these calcium will either bind to the calmodulin protein specifically so that the calmodulin protein can interact with some of the kinases and activate the downstream effect molecules. Or this calcium ions and DAG are able to activate some of the protein kinase C. So this protein kinase C is able to phosphorylate some of the transcription factors. So this transcription factor will go into the nucleus and activate some of the gene transcription. So that we have discussed yesterday. In today's class, we will discuss about these two remaining pathways. One is PI3 kinase pathway and second is RAS-MAP kinase pathway. So actually, I want to discuss RAS-MAP kinase pathway first, but actually in Cooper, this PI3 kinase pathway is given earlier, and then we will come to RAS-MAP kinase pathway. Okay, this PI3 kinase pathway is also common in mTOR pathway, so it is given here. So this is called PI3 kinase AKT pathway and mTOR pathway. This is completely different pathway, but this is single pathway. What is this AKT? Now the question is, the AKT is actually the protein kinase B. I was telling you about this particular protein kinase B. So the other name of protein kinase B is AKT. And this particular AKT is a protein serine threonine kinase. Okay, so you need to remember and need to underline this, that this AKT, first of all, this is a protein kinase B, PKB, and it is a serine threonine kinase. So it is either phosphorylating either serine or threonine. Okay, so these two things you need to remember from this particular discussion. Now, yesterday I told you that the two type of secondary messengers will result when this breakdown, this particular bond is broken down by the phospholipase C, beta or gamma. Beta in the case of GPCR and gamma in the case of receptor tyrosine kinases. Now, after breakage of this bond, the two type of secondary messengers will result. One is IP3 and one is DAG, disylglycerol. IP3 is a small polar molecule, so will go into the cytosol and will able to bind to the ryanodine receptors, so that these can come into the cytosol. IP3 is a small polar molecule in the cytosol. It will bind to the plasma membrane receptors for calcium ions, and will open those calcium ion channels, so that the calcium ion concentration within the cytosol will increase, or it is increased, and calcium ion concentration will able to activate some of the calmodulin protein or some inactive protein kinases. Okay. Then the second possibility is this PIP2 with the help of some enzyme called PI3 kinase, because it is phosphorylating this particular PIP2 at third position, so converting it into PIP3. And this PIP3 is another secondary messenger. Okay, so PIP2 not only serves as a source of diacylglycerol and IP3 as we have studied yesterday, but it is also the starting point of distinct second messenger pathway that plays a key role in regulating the cell growth and survival. In this particular pathway, PIP2 is phosphorylated over the third position of inositol by an enzyme called PI. PI is phosphatidyl inositol 3 kinase. So it is a kinase, so it will add gamma phosphate from ATP into this third position of inositol. Like phospholipase C, one form of PI3 kinase is activated by G protein, while the second form is activated. A second form has an SH2 domain. Okay, so someone is asking that what is the function of these proteins? So the protein which is having SH2 domain will interact with phosphotyrosine, and the phosphotyrosine interacting protein will be activated, and it will now convert this PIP2 into PIP3. Okay, and activated by associated with the receptor protein tyrosine kinases. So phosphorylation of PIP2 yield a second messenger that is called phosphatidyl inositol 3,4,5 triphosphate, PIP3. Now, this key target of PIP3, which is critical for signaling the cell proliferation and survival, is a protein serine threonine kinase that is called AKT. Now, come to this particular diagram and see here. Okay, so this is what will happen when this PIP2 is converted into PIP3. So it will interact with another protein kinase B. This AKT you can write here. This is a protein kinase B. So this AKT is a protein kinase B. And what is the special thing about this protein kinase B is this is having a pH domain. What is pH domain? This is not your common pH, acid base, but this pH domain is some plextrin homology domain. Okay, so something it is discussed somewhere in the text. Now, what is the first showing you this pH domain? So this is plextrin homology domain. So this pH domain means some plextrin homology domain. So what is the importance of this particular domain? That whenever this pH domain is present, okay, so this AKT is recruited to the plasma membrane. AKT is, that is protein kinase B, is generally present in the cytosol, but will be recruited or localized to the plasma membrane and is able to bind to the PIP3 via this pH domain. This pH domain helping, okay, activated by the result of phosphorylation of another protein kinase PTK1 that is bind to PIP3 as well as this protein. So it is not given here, but it is given somewhere. Okay, I'm telling you. So what is the importance of this pH domain? So the proteins which are having this pH domain, for example, this protein kinase B or AKT protein, or this PDK1 protein kinase, this PDK1 is having this pH domain. So they are able to bind this PIP3. So if it is having 3 phosphates over 3, 4, 5 position, they are able to interact and they are able to localize over this plasma membrane. So it will help, this pH domain will help in the interaction with PIP3 in a similar way like SH2 was helping with the interaction with the phosphotyrosine, for example. Okay, so see, A key target of PIP3, which is critical for cell signal proliferation, is AKT. PIP3 binds to the AKT known as plextrin homology domain. This interaction recruits AKT to the inner face of the plasma membrane. Okay, so this is plextrin homology domain, and interaction recruits the AKT to the inner face of the plasma membrane because it is able to bind with PIP3. Where it is phosphorylated, it is phosphorylated. So this protein kinase B first being phosphorylated over its tyrosine residue, activated by another protein kinase called PTK1. This particular kinase will come and phosphorylate the AKT or protein kinase B. Then this AKT or protein kinase B contain a plextrin homology domain bind to PIP3. Formation of PIP3 thus result in the association of both AKT and PDK1. Okay. With the plasma membrane. So they are nearby, leading to the phosphorylation and activation of finally this AKT. Activation of AKT also require phosphorylation at the second site by the distinct protein kinase. So at certain position, this PDK1 will phosphorylate. At other position, some other protein kinase will phosphorylate, which has recently been identified as a form of mTOR complex with. So it was not identified, only recently it is being recognized that mTOR is another protein kinase which will activate the AKT actually, or this protein kinase B over the second position. So complex with protein called Richter protein. So there is another protein complex. So mTOR is forming a complex with Richter, and it will phosphorylate AKT so that it can be activated. The mTOR Richter complex is itself stimulated by some growth factors, but its mechanism of activation remains to be understood. So we do not know about the mechanism, how it is activated, but it is known that this mTOR is forming a complex with some Richter protein. So see here, one phosphorylation is done by this mTOR and Richter complex. One phosphorylation is done by PDK1 complex with the complex with this plasma membrane. And then finally it will activate. When the two phosphorylation will happen, the AKT will be phosphorylated and activated. And then it will activate other proteins like BAD. It will activate some transcription factors. It will inhibit GSK3 beta as we have studied earlier, GSK growth factor. Sorry, we have not studied about GSK, okay. So it will inhibit GSK3 beta, and it will also inhibit one of the protein that is called, so it will also inhibit one of the protein that is called FOXO. So you can add one more thing here, that it is inhibiting GSK3 beta. It will also inhibit one protein, important protein that is called FOXO, FOXO. I will let you know what is FOXO and these things, okay. So first of all, it will inhibit. This is an inhibitory sign, not as arrow. So it will inhibit FOXO also. It will inhibit GSK3 beta, but it will activate some of the proteins. So that's how it will activating or inhibiting some of the downstream effector molecules. So I think that is enough for today's class. Okay, so we will start our discussion from this particular AKT only, that is protein kinase B. Okay, and how it is activated we have seen. Now we need to see what is the possible ways of interaction, how once after activated this AKT phosphorylate the number of target proteins. So these target proteins are actually discussed here, FOXO, some of the cytosolic saffron protein 1433. Okay, so these are GSK3 beta or some of the other proteins, for example. So we will start our discussion from this particular section in tomorrow's class, and we will finish this first. Then we will discuss about RAS MAP kinase pathway. Okay, so this RAS MAP kinase pathway will remain, and IP3 DAG pathway as we have studied earlier. So this IP3 DAG pathway is also given a short note here that that was discussed earlier, and it is including phospholipase C gamma. Okay, so these things I already told you in yesterday's class. This is, we are studying actually PI3 kinase pathway, and after finishing this all things are given here actually. The docking site for PDK1 and protein kinase B. This protein kinase B is AKT actually. Okay, it is written here. This PKB is a AKT actually present in the cytosol, and then will come to the plasma membrane. Okay, so it will. Then we will study something about insulin signaling pathway, because this is very important. And we will finish our discussion of MAP kinase pathway. After MAP kinase, we are having, sorry, this came again. We are discussing this mTOR pathway and JAK-STAT pathway, and some other pathways which are remaining actually in your book. Okay, so it is a time for queries, and only 4 participants are. Oh my. So okay, so there is no problem. If you face any query or problem when you read this text, you can directly WhatsApp me. Okay, so Shalu is having no queries. If you have any, when you read the book or text, you can directly send me WhatsApp. Okay, so I will see, and we will discuss in tomorrow's class. No problem. Okay, so bye bye. Take care. Good night.